I'm Vicky Bradley, and today I'm going to be talking about human immunity, covering the three main topics of non-specific defences, specific immunity and the biology of vaccination. We live in a world surrounded by microorganisms. Some of these, known as pathogens, can be harmful to our bodies by causing infection and disease. To protect us from these disease-causing pathogens, we have the immune system. Infection is caused when one of these pathogens enters the body and multiply either by infecting a host and reproducing themselves or by replicating themselves, for example, in the case of a virus. A virus is a small organism that is not considered to be living. It finds a host and then replicates itself. Bacteria are single-celled organisms, a little larger than viruses, that cause infections such as strep throat or meningitis. Fungi cause many skin diseases, including athlete's foot, and parasites cause infection using a vector, for example when a mosquito bites a human and infects them with malaria. Take a look at handout 1, also seen on the screen. These are examples of different types of pathogen and how they are transmitted. A common way infection can be transmitted is through direct contact. This is when an infected person is in physical contact with an uninfected person, for example kissing, sexual contact or contact with bodily fluids. This can also occur from mother to unborn child via the umbilical cord. Indirect contact can occur when there's no direct contact between the infected and uninfected person. For example, if an infected person coughs or sneezes, leaving particles in the air or on an object that is later touched by an uninfected person. Another form of indirect contact is when a vector carries the pathogen onto an uninfected person, for example in the case of mosquitoes with malaria. And finally, contamination can happen by drinking water or eating foods contaminated with the pathogen, for example E. coli, which can be found in undercooked meat or unwashed salads. The body has three lines of defence against infection. The first two are non-specific, which means they don't distinguish between different pathogen types, responding to them all in the same ways. These are defences we're born with, and they can be seen as the first or second line of defence. The first line are physical and chemical barriers that prevent the pathogen from entering the body. For example, the skin, which is the largest organ in the body. As long as it remains unbroken, it will remain waterproof and prevent microbial organisms, toxins and chemical irritants entering. Another way to enter the body is through mucous membranes. These contain enzymes to help flush away any external organisms entering the body, and they can be found in the ear, nose and throat, and the intestinal, respiratory, urogenital and vaginal tracts. In the eyes, the enzyme lysozyme, which can be found in tears, flushes away and fights bacteria. Cilia are tiny hairs that protect the cells in the airways. They trap and remove foreign particles and bacteria from the internal surfaces of the body. Enzymes in the stomach kill any pathogens before they can cause an infection, and urine flow flushes pathogens out of the bladder and urethra, preventing UTIs. If a pathogen manages to enter the body, the second line of defence begins to respond. Usually this starts with an increase in body temperature to slow down or destroy pathogens. At the site where the pathogen has entered, inflammation occurs as blood flow is directed there, causing dilation of blood vessels. Cells around the site develop gaps, allowing more white blood cells to reach the area. Different cells arrange themselves along with proteins known as cytokines, which fuel and enhance the immune response. Heat will occur, causing an antibiotic effect, and tissues appear red, swollen, and feel painful due to pressure on the nerve cells. A type of white blood cell called a phagocyte begins a process called phagocytosis. The phagocytes become specialised to fight that specific infection by engulfing the pathogen and then destroying it with enzymes. Macrophages make proteins that either pierce holes in the bacteria and parasites or stick to the outside of the bacteria, attracting the phagocytes to them. The third line of defence is known as acquired immunity, and this develops after a person is born and throughout their life. This involves activation of cells to become specific to the pathogen they are attacking and remembering it if it enters again. Each antigen has a unique shape which the immune system learns to respond to, and so its response becomes specific. Active immunity can be active, passive, and attained naturally or artificially, which I'll cover in more detail later. Leukocytes are white blood cells that are produced in bone marrow. They come in 5 types, each with different roles. Neutrophils are the most common and the first to arrive at the infected site. They attach to the walls of blood vessels, preventing germs entering through cuts, and are responsible for beginning the process of phagocytosis. They kill bacteria, fungi, and anything foreign that enters the body. Monocytes are the largest of the white blood cells. They complete phagocytosis by developing into macrophages and eating up dead cells. Eosinophils are cells which release toxins to kill pathogens from parasites and worms, and they can also develop in response to some cancers. Basophils are the white blood cells that are least prevalent in the blood. They use histamine to dilate blood vessels, allowing other immune cells to reach the infection site more quickly. Lymphocytes are the cells that play an important role in antibody production. They are present in lymphatic tissue such as tonsils, spleen, and lymph nodes. They produce antibodies and create memory cells which remember a specific pathogen, which I'm going to cover in more detail now. Of all the leukocytes, lymphocytes are the biggest in numbers. There are millions of them travelling around the body in search of foreign cells to attack. The three main types of lymphocytes are T cells, B cells, and natural killer cells. Natural killer cells recognise immune stress and kill many types of tumour and virus-infected cells without the need for exposure to an antigen. T and B cells work by recognising and responding to antigens. T cells mature in the thymus gland and make up around 80% of lymphocytes. They kill enemy cells and trigger the activation of B cells, which multiply and produce specific antibodies. This is done with the help of antigen-presenting cells. They present a small amount of the pathogen to the T cells, and they become activated, and different types of T cell are then released in the bloodstream. Helper T cells release signals in the area around the antigen, attracting macrophages who then destroy by phagocytosis of the invading cells. Cytotoxic T cells bind to antigens and release a poison specific to that antigen. They attach to the antigen and cause the cell to self-destruct, avoiding the escape of viral particles. And finally, suppressor or regulatory T cells control the helper and cytotoxic cells to avoid autoimmune attacks on the body's own cells. The B cell's main purpose is to recognise antigens and produce antibodies specific to them. They present the antigen to the T cells to stimulate a response. They mature in bone marrow and then migrate to lymph nodes, liver and spleen. When they're activated by the T cells, they divide rapidly, creating clones of plasma cells that produce millions of antibodies specific to that antigen. These are then used to attract macrophages which kill and digest the pathogen, dying themselves after the primary immune response. They also produce another cell when they are cloned called memory cells. These cells remember the antigen and travel to lymph nodes where they remain ready to attack more quickly if that specific antigen enters the body again. This is known as the secondary immune response. Immunoglobulins or antibodies are Y-shaped protein molecules that bind to antigens on pathogens and any foreign materials that enter the body. They're made up of two heavier chains shown in blue and the two lighter chains shown in red. At the end of each arm there are receptors that identify foreign bodies. Their role in the body includes targeting infection and recruiting immune cells and other cells that eat external substances. They neutralise toxins and activate mast cells to protect from parasites. The different types of antigen are shown on the screen. IgA, which is found in mucosal tissues such as the mouth, vagina or intestines. IgD, which acts as an early immune response. It's produced by B cells as a signalling antibody to trigger the release of IgM antibodies. IgE, which is released by the lymph nodes and binds to an antigen, causing the release of histamines from basophils and mast cells to protect the body from parasitic infections such as worms. IgG, which are the most abundant in the body, they stimulate an attack on anything foreign they encounter in the body. They're the only antibody that is passed from the mother to the baby from birth, protecting the baby from any infections it has not yet been exposed to. And finally, IgM, which is structured differently in that there are 5 Y-shaped immunoglobulins attached at the base. It's one of the first antibodies to respond, but is then taken over by IgG antibodies. IgM are important in forming B-cell memory cells, enabling the body to respond more quickly the next time it is exposed to the same pathogen. As antigens are present on all cells, it's important to remember that the immune system recognises cells that are self and non-self to avoid attacking all of the cells in the body and to respond when foreign cells are identified. The level of response from the immune system is stronger if the antigen is very different from the cells. For example, if the cell is not a human cell, it will cause the strongest response. To help the immune system recognise the differences, it uses the major histocompatibility complexes 1 and 2, along with the leukocyte gene complex. MHC1 is found on the surface of all nucleated cells. It extracts fragments from foreign proteins if they are found and presents them to T cells. Similarly, MHC2 also presents fragments, but only from extracellular pathogens such as bacteria. The fragments are digested with lysosomes after they have been endocytosed by B cells and phagocytes. Hypersensitivity occurs when there is an inappropriate response of the immune system. This is usually in the form of an allergy, autoimmunity, or alloimmunity. When self-tolerance fails, this can cause an excessive or inappropriate response to self-antigens. When cell types are being produced, self-reacting cells are destroyed. Sometimes this process fails and causes T and B cells to be activated. The antibodies produced during this activation can cause a response against the body's own cells. Hypersensitivity diseases come in 4 categories, as you can see on the screen. Type 1, immediate hypersensitivity. Type 2, antibody-mediated hypersensitivity. Type 3, immune complex-mediated hypersensitivity. And Type 4, cell-mediated hypersensitivity. A problem when transplanting organs is the immune system's rejection of non-self cells and tissues. Chances of rejection can be reduced by closely matching organs and donors to the recipient. When they're not matched closely enough, the blood transfusion or organ rejection can occur. The closer the match, the less likely the organ will be rejected. Tissue typing is done to test lymphocytes for human leukocyte antigens. To check compatibility, and monoclonal antibodies are used to prevent T cells rejecting the organs and destroying the tissues. There are 3 types of rejection that can happen when transplanting organs. Hyperacute, which happens instantly, and the recipient can die if that organ isn't removed. Acute rejection, which happens up to 3 months after the transplant. Or chronic, which can happen over a long period of time and involves the body's own immune system constantly attacking the new organ. Drugs such as cyclosporin can inhibit the production of MHC2, which prevents activation of T and B cells, thus suppressing an immune response. There are over 80 autoimmune diseases. These occur when the body mistakenly attacks self-antigens. It's believed that triggers include bacteria, viruses, drugs, chemical irritants, and environmental irritants, and they tend to be hereditary. Usually symptoms include fever, fatigue, muscle aches, and inflammation. Sometimes symptoms disappear, but they can flare up usually when they're triggered by one of the aforementioned factors. 2 examples of autoimmune diseases are rheumatoid arthritis and muscular sclerosis. Rheumatoid arthritis causes chronic inflammation and happens when the body's immune system attacks the cells lining the joints of the body. This causes painful swelling and eventually bone erosion and joint deformity. It's more prevalent in women and is more likely if a person smokes or has a family history of rheumatoid arthritis. There is no cure, but pain can be managed and progression can be slowed with medication. Physiotherapy can also help to retain mobility, and surgery can be given if joint problems develop. MS is caused by the immune system attacking the myelin sheath. This is the outer layer surrounding and protecting the nerves. When this becomes damaged, messages become slowed and distorted. This causes a range of problems including mobility, stiffness and spasms in the muscles, fatigue, vision, balance and cognitive problems. Like rheumatoid arthritis, there's no cure, but treatment can be given in the form of steroids to speed up recovery from a flare-up. Disease-modifying therapies help reduce the damage and scarring of the sheath, which is thought to reduce relapses and slow worsening disability. Other medications are given to sufferers to help with some symptoms such as fatigue and visual problems, and physiotherapy helps with muscle spasms and stiffness. As mentioned earlier, acquired immunity is developed after birth and throughout a person's lifetime. The categories can be divided into active or passive, and further depending on whether the immunity is acquired naturally or artificially. Active immunity occurs when the individual's own immune system responds to a foreign substance, and passive is when immunity has been transferred from another person. Natural passive immunity happens when antibodies are transferred from one human to another in a natural way, for example after birth when breastfeeding. This provides protection for a short period of time until the child's own immune system develops. Active passive immunity offers protection for a short period and is when antibodies have been developed by another person and are transferred by a blood transfusion or injection. Natural active immunity occurs when the body is exposed to a pathogen and creates antibodies in response to the infection. If you were exposed a second time to the same pathogen, the body will respond quicker as the memory cells would have remembered the antigen from the previous exposure. Artificial active immunity happens when exposure is deliberate, in the form of a vaccine, when a small amount of the infection is injected, an immune response is triggered, creating the production of antibodies and memory cells to remember the infectious agent if it is encountered again. Artificial interventions come with a risk of side effects, with passive side effects being more severe. In both types of passive immunity, the person's own body is not involved in creating the antibodies. And though immunity is immediate, it does not last a long time. Whereas active immunity can take time to develop, but usually lasts longer than passive, sometimes a lifetime. A vaccine is a substance which contains dead and harmless fragments of a pathogen, along with toxins produced by the pathogen and live pathogens which have been treated to make them harmless. The body reacts to this exposure by stimulating white blood cells which produce antibodies specific to the pathogen. Although the vaccine is considered harmless and does not cause a risk of developing the disease or infection, the initial exposure can trigger mild side effects. If the person is later exposed to the same pathogen, white blood cells are produced more rapidly, destroying the pathogen more quickly through immunological memory. After the vaccine, the initial immune response is triggered, and over a period of around two weeks, the response reaches its peak, enabling memory cells to be produced and remember the pathogen if it's encountered again. If the pathogen is encountered again, the response is much quicker and stronger in response due to the abundance of memory cells already present in the body. There are a number of different types of vaccines used to prevent disease and infection. Live vaccines come from weakened versions of the virus or bacteria, which is done in a laboratory. Then small doses are given to produce the immune response, which involves replication within the body. This causes the response to be almost the same as if the infection had been encountered naturally. Inactivated vaccines are from bacteria or vaccines which have been developed in a culture medium and then inactivated using heat or chemicals. As they are not live, they cannot replicate or cause disease from infection. Multiple doses are needed, with the first dose preparing the immune system and subsequent doses providing a stronger response and more protection each time. Vaccine duration differs depending on the type of vaccine and the disease or infection it's protecting. If you take a look on your handout, which I'm going to bring up on the next slide, you can see the different types and the duration that they protect for. Some vaccines can last a lifetime, such as measles, whereas some are shorter, but still relatively long, requiring a booster to maintain immunity. With diseases that depend on transmission from one person to another, immunisation is not only beneficial to the vaccinated individual, but also to the wider community, as it reduces the reproduction rate, and in some cases the disease completely dies out. If we look at the slide, we can see how contagious disease spreads rapidly throughout the population when no one is immunised, how it spreads moderately when some of the population are immunised, and how very few people are infected when the majority of the population are immunised. In the case of the latter, the disease finds it difficult to spread, resulting in containment of the disease. This is known as herd or community immunity, and it means that those in the community who cannot be vaccinated, for example people who are vulnerable, elderly or newborn babies, have some level of protection if the disease is less prevalent. Due to the success of widespread global vaccination programs, smallpox became the first contagious disease to be eradicated in 1980. Herd immunity doesn't work with all diseases. Tetanus, for example, is not caused through transmission between people. This means that the individual needs the vaccine to be protected, as it is not caught from the people surrounding them. Herd immunity is affected by a number of factors, including different strains of an infection if the pathogen mutates, people with compromised immune systems not responding to the vaccine, and some populations choosing not to take the vaccine due to religious or ethical reasons. There are growing numbers of people who believe that some vaccines are harmful. These numbers have increased since the COVID-19 pandemic and more people accessing social media. In 2020, The Lancet reported that there are 31 million people on Facebook following anti-vaccine movements, a number which increased by almost 8 million since 2019. The benefits of vaccinations are clear if we use smallpox as an example. Until the 1970s, the World Health Organization estimated that there were 50 million cases a year, and of those, 1 in 3 died, mostly under the age of 10. A disease that caused such high numbers of child mortality no longer existing is surely a good thing. And this is not isolated to smallpox. With vaccines for many other diseases that have prevented millions of deaths, the numbers speak for themselves. Other benefits include cancer prevention, with the HPV and hepatitis B vaccines reducing associated cancers of the cervix and liver, increased life expectancy, and a reduction in people developing antibiotic resistance due to lower levels of bacterial infections. The social and economic benefits worldwide are also clear. By keeping adults and children in society healthy, this reduces time lost from school or work, resulting in more healthy children who perform better at school and a stronger, healthier workforce of adults who lose less days to sickness. Thank you for listening.